Right, thank you very much, Hamish, for giving me an opportunity to talk here. My name is Benjamin Schuster Böckler. I'm at the Ludwig Institute for Cancer Research in Oxford, and my group really is working on computational biology, and since the last few years, specifically on trying to find ways to detect cancer early from cell-free DNA. I'm thereby relatively new to the liver field, and my main connection to that is through Ellie Barnes and the DELIVER trial, which I'll talk a little bit about it. So, forgive me for not being quite as steeped in on the medical side of things and the liver side of things than some of the other people today. There will also be quite a bit of talk about case control studies, for which after this talk I apologize in advance. But that is what we have at the moment. So briefly, for those who don't know yet, or if you've lived under a rock for the last 5 years or so and you've never heard of cell-free DNA, in principle, the idea is that most cells, when they die, either through apoptosis, necrosis, or some other pathway, they will release some fragments of DNA into the bloodstream. That DNA has about a half-life of a few minutes, will get cleared, but you can extract it from plasma. And cancer cells will also shed DNA into the blood, sometimes more, sometimes less. Why that is, it's not so clear, but they do. And when they do shed DNA into the blood, that will contain some of the features that are characteristic for the tumor, such as point mutations, copy number variations, rearrangements in the genome, but also epigenetic features. So really, these genomic and epigenetic features can be extracted from sequencing that cell-free DNA and could be used to detect cancer and hopefully detect it early. So since this idea has really sort of gotten into people's head, there has been an incredible tidal wave of interest in using cell-free diagnostics through sequencing of cell-free DNA. And there's a that is not even all of them. There's like a gazillion companies now doing that sort of thing. And these companies have different perspectives. Some of them are trying to do very much prognostic assays for specific cancer types. Others do diagnostics for very specific cancer types. And yet others are trying to do what we call multi-cancer early detection, MCED. And they also have different ways of going about this problem and how to solve it. So what kind of features you use from the blood that might be useful. So I'm going to just briefly talk about the ones that are most advanced by now in the kind of early cancer detection setting. And almost all of these companies, here's just a sort of a list of some of the bigger ones, almost all of them use an epigenetic assay, or an assay that at least to some extent uses epigenetic features in DNA in their setting, in their assay setup. So why is it that epigenetics is so interesting in cell-free diagnostics? So the reason is really that mutations are extremely heterogeneous across patients. So if we just try to find any two patients that had the same mutation in any of their driver genes or anywhere else in the genome, we would probably be out of luck. That's extremely unlikely. This is from the liver cancer TCGA paper, where they show which mutations do different patients have. And you can sort of make out that really it's really scattered across. Copy number is a little bit better, but it's still not a kind of unifying feature. So on the other hand, DNA methylation, as shown in this picture, where basically here what's shown is the methylation state at various places in the genome in all the patients from that TCGA liver cancer cohort. And on the left and the right is sort of normal control blood and normal liver. And so what you see is that the cancers do tend to have quite consistent methylation in certain areas where there is no methylation in the blood or in normal liver. And this is much more consistent between individual patients. So there is kind of a sense that this kind of epigenetic map is more likely to yield a good biomarker for early detection. So the first people to really take this forward into a large trial were a company called Grail, and their test is called Gallery, which, as you probably know, is also being trialed in the UK in various also prospective trials. But this is from a case control trial that they ran some years ago, where they took a number of different cancer types and collected patients and controls for each of them. And they reported very kind of high accuracy numbers for all of those, for a lot of the especially the solid tumors there. And so here's the liver cancer kind of, I'm just highlighting it here and showing sort of what they claim is their sensitivity at 95, what was it? I think at 90% specificity or something. I don't remember now exactly. And they kind of suggested that they get incredibly good accuracy of detection using the Grail, using the Gallery assay. It should be noted, though, that I'm pretty sure that their controls in this case were probably not cirrhotic patients. They don't really say, but I have my doubts, which makes this somewhat an easier problem. And the numbers are quite low. So I don't know what exactly they mean with stage 1 here, but there is only 6 patients. So nevertheless, it's encouraging. There's another company which very recently reported results only really late last year, a company called Delphi, not to be confused with our DELIVER study sub-cohort called Delphi, spelled with a PH. So Delphi is a company founded by Victor Velculescu in the States, and they use a different, sorry, I should have said Grail uses methylation, a methylation-based assay. Delphi here, they use a different assay which focuses on chromatin conformation, which you can measure by looking at where the DNA in the blood broke off, and you can sort of make some statistics about the distribution of these breakpoints and about kind of the length of the fragments that you find in the DNA. And using that assay, they show this beautiful picture which suggests that you get kind of progressively more dysregulated patterns of this fragmentation from non-cancer, no liver disease to cirrhosis to HCC. And they use this kind of pattern to create a single score. I'm not entirely sure how, but they basically condense this into a single number. And this is what they reported as sort of the distribution of that number in patients with hepatitis, cirrhosis, and then cancer. Again, the numbers are not very large, and again, this is a case control cohort. At least in this case, they do report cirrhosis directly, and I don't know if you can make it out. I can't actually see very much on this screen, but the dots are kind of very, very, very faint. But on the one hand, if you just look at the median and the distribution, it looks quite promising. They clearly do separate the tumors from the cirrhosis, but you can also tell that there is a fair number of cancers with low Delphi scores. So I think they don't actually report the sensitivity specificity in a way that I was happy with, so I didn't put it on the slide. But I think you can just visually make out that there's a fairly large number of cases which weren't picked up. So sensitivity is not perfect, and mainly specificity is not very good at all. Like cirrhosis, there's a lot of cirrhotic patients that clearly are within the same range of this Delphi score as the tumors, especially the early cancers. So still work to be done here in method development to figure out how can we improve sensitivity and specificity of detection of HCC from blood against a cirrhotic background. So Michael mentioned it earlier. I'm also part of the team that of the DELIVER team. There is in DELIVER 2 components. One is again, case control cohort, or rather 2 actually, Selena and Delphi. But to keep things simple, the key point about these case control cohorts is that they allow us, as was discussed earlier, to really come up with putative biomarkers that we think are at least worth testing, so that we can then take them forward in a prospective cohort. I don't think we want to start from the prospective cohort and then do another one just to kind of validate it. So what we're doing here is we are sampling both blood from cases and controls, so all like cirrhotic, but no cancer, and cirrhosis and cancer, from patients with a number of different diagnoses. And we're also taking tissue samples, which is very important for us because on the computational side, what we're really trying to do is we're trying to understand what is the biology of the disease, what is the epigenetic landscape of tumor and cirrhosis that would allow us to not just sort of go blind and requiring a gigantic number of patients to identify the markers kind of just from the blood itself, but we're trying to sort of find some regions that are really clearly discriminatory and then take those forward into a test. And then once we kind of have a good molecular biomarker, hopefully, and maybe also image biomarkers, we can take this into this prospective cohort that Michael mentioned earlier, which is still ongoing. So I think we're sort of about halfway through collecting. The goal is to collect about 3,000, to recruit about 3,000 patients and follow them for several years. And so we will, I hope, have at least 3 blood samples. So if there is a diagnosis at any point, we should be able to go back, and it should be hopefully no more than a year before the diagnosis that we have a blood sample. So we can then really see, we can test our markers on that blood. So what makes our approach a little bit different is that we have an interesting new methodology called TAPS, which was developed by the lab of Xunshan Song in Oxford. He's also part of the Deliver team. And my group did all the kind of computational analysis. And I'm not going to bore you with the magical chemistry, but it's a method that can detect DNA methylation in a much more sensitive way than bisulfite sequencing. And what I mean with sensitive is that when you apply it to the kind of very low volume DNA that you get from a single blood draw, it doesn't degrade the DNA, so you get more information out just by losing less DNA. But the key point is that what I'm showing here is on the top is kind of how the landscape of reads looks like if you do bisulfite sequencing. At the bottom is what you would get if you do TAPS. And the colors basically denote bases that were changed. At the bottom, you see there's almost no changes, which means that this looks like normal whole genome sequencing. You can detect mutations. You get very relatively even coverage, just like you would from normal whole genome sequencing, but you also get only in certain positions you get information about the methylation, whereas bisulfite sequencing just sort of like changes all the DNA into C and into T and A, and also causes like massive variation in coverage, which basically makes it not very suitable for the kind of assays like the Delphi or any kind of mutational assay that we think would be useful to develop a biomarker. And so the goal is really to take this kind of methodology and extract not just one but multiple features, methylation being probably one of the most important within them, but we could also look at copy number, we could look at specific mutations, we could look at fragmentation patterns and so on, and train models on that kind of combination of information and take that forward. And in like a very small, again, small case control study that Xunshan initiated a few years ago, we could tell that against controls, we were actually able to identify liver cancer with quite good accuracy. But again, I wouldn't read too much into it because I do agree this is a small cohort and not perfectly set up. But we're really close now to actually taking this forward to the much larger patient set that we have through the liver. And with that, I'll thank mainly the Deliver team for making this amazing effort to putting these cohorts together, people in my lab and in Xunshan's lab for doing all of the lab work and the computational work, and my sponsors and various people who are involved. There is other companies and research organizations that are involved in funding this, so thank you.